All right. It's all good. So, today, we're starting cell chemistry. We're starting cell chemistry. It's exciting. So, I believe the course code is cell chemistry. CCS24-1. What's the name of this here note? Cell, scale, and structure. Ooh. Cell, scale, and structure. So, what we're doing today is we're starting to put together some of the stuff that we learned in the last two sections, in the introductory biology section, in the introductory chemistry section or unit or whatever you want to call it. And what we started to figure out was sort of, some of the basic aspects of cells and how they structure and function, some of the basic aspects of chemical molecules and how they structure and function. Now we're going to mash those two things together and start talking about first a refresher of basic cell structure, but we're going to start getting closer and closer to the individual parts of these cells and start figuring out where their chemical bits start and begin and end, and how that all wraps together into the beautiful whole that is us, man. Woo, crazy. All right, let's do this. So, cell, scale, and structure. Click my compose button. Beautiful. So, the major place that we're getting these results is from electron microscopy, microscopy, right? Electron microscopy, I'm going to stop trying to say it. This technique of taking images of things is really, really cool. We can talk about it later, but the basic premise is that if we're taking images of things with light, light has a particular limitation on its wavelength. It can only get so small, whereas electrons can get even smaller wavelengths than light. Roughly speaking, the size of the wavelength dictates the size of the thing that can be imaged, so smaller and smaller wavelengths allow you to image smaller and smaller things. Therefore, you're taking images using electrons, you get to take pictures of smaller stuff than if you're using photons. That's the basic premise. That all being said, for us, what matters is that it just takes really cool pictures of small stuff that we can't see in other ways. Most of these cells have been captured or preserved using a number of different techniques. In the old days, our classic way was to turn everything into crystals and then take pictures of them from there. But we've gotten much more advanced since then, which is nice because it means we don't have to analyze crystal. Don't have to analyze crystals quite as much anymore. So, if we're looking at images, we need a yardstick. We need a way to measure them in order for it to actually be science. So how do we measure things that are really small? Ooh, this is a small one. How do we measure things that are really, really small? So atoms are measured in terms of nanometers, which is a very small quantity. In terms of nanometers. What is a nanometer? Size of the atom. 1 times 10 to the 0 equals 1 nanometer. This is also 0.001 micrometers. And one of your questions might be, nanometer, how small is this in relation to an actual meter? Stay tuned. Maybe I'll tell you, maybe I won't. Proteins. Proteins are measured in terms of tens. Is that right? It is. Tens of nanometers. This actually gives us sort of a very straightforward kind of relationship, which is kind of cool. If atoms are about nanometers yay big, and notice that atoms can have quite a large range, but if atoms are about yay nanometers big and proteins are tens of nanometers, well, that means that we can assemble like 10 atoms together and start to build a protein. Ooh, interesting. Yeah, and that's not totally wrong. Notice that we're dealing with tens plural, though. So it's actually much more likely that we're going to get proteins when we're dealing not with 10 atoms itself, but more like 30 or 40 or 50 atoms, which is where our sort of average range is being implied to be here. Right? We're dealing with tens. That gives us a range from 10 all the way to 100. We don't want to stay focused on the very bottom of that range. Right? Simple stuff. So, what color shall I take? Proteins are measured in terms of tens of nanometers. This is 1 times 10 to the 1, right? Which equals 10 nanometers. And this, if we reduce our value over here, gives us 0.01 micrometers. Micrometers and nanometers can be used to measure the same things, but we use different words for different sizes, basically out of convenience. You know, if we can talk about something being at 10 nanometers as opposed to being 0.01 micrometers, I'd rather say 10 nanometers than 0.01 micrometers all the time. 0.01 micrometers, 0.01 micrometers, right? You know, it's just convenience. And that's very important when you're dealing with really radically large different scales of things. So, viruses, proteins are tens of nanometers. Viruses are hundreds of nanometers. Start making this list move a little more quickly. Viruses are hundreds of nanometers. Bacteria, you might be interested to know, are thousands of nanometers. So we can think, and you should think, of many viruses as preying on bacteria. There's an entire particular class of bacteria plays on. There's an entire particular class of virus, that preys on bacteria called phages. And these are really important viruses to understand, and we'll talk about them later. And viruses, therefore, are much smaller than the bacteria that they're attacking. Right? We can think of them as kind of like little animals ganging up on a big animal, same way as we think of humans ganging up on mammoths or a pack of dogs ganging up on any number of big creatures, right? Same sort of deal. So bacteria are thousands of nanometers. Red blood cells are tens of thousands of nanometers. And I think this is really cool as a relative scale, because red blood cells are complicated and they're common and they're a really big important part of us. And you can see how, in terms of a very regulated ecosystem, cellular ecosystem like you are, like your body is, we can produce these really large complicated cells that we have to have essentially other cells around to take care of. Red blood cells provide this one very specific function, but other than that, that's what they do. Does that kind of make sense? You know, they don't provide the other necessary things for life all by themselves. They require all of these extra supports that the body gives them. And if you take red blood cells out of our natural environment, then they stop making sense as cells, and they cease to function. Does that kind of make sense? I'll come back to that later. Let's get my pink again. I got a nice pattern going on here. So, last but not least, your hair. There's actually a lot of variety, but it's about 100,000 nanometers. About 100,000 nanometers across. So the width of a hair. That's kind of cool and kind of crazy. That means like blood cells, you know, they're taking place across veins that are at least the size of your hair, if not bigger and sometimes smaller than your hair, but they're in that sort of range. And so you get this idea of these crazy different scales. You can see veins and stuff inside of your body, right? So it's cool to start to, and you can see hair, right? So you could start to like make relative relationships between these different things and start to fit together just how small the virus is, just how small the actual cells that we're dealing with here is. So for us, we're going to be dealing very much with this sort of 10,000 to hundreds of nanometers section, which of course actually puts us in much more comfortable range, as you will notice from my micrometer talk here. To be talking about more about micrometers a lot of the time. So if we're dealing with viruses here, 1 times 10 to the 2, this pen's getting sad, let's try U. 1 times 10 to the 2, that's okay, but we're just going to keep throwing things at it until we get perfection. Exactly. That's marginally superior. We'll take that. At 1 times, see how quickly my standards drop. 1 times 10 to the 2 equals 100 nanometers, or of course, 0.1 micrometer. Close enough. And then over here in the thousands of nanometers, 1,000 nanometers is a micrometer. So this is really cool, and this sort of describes the reason why we're dealing with these different ranges. When we're talking about overall sizes of bacteria and cells and things, we're often going to want to be dealing with micrometers, just because this is like a 10 micrometer size meter. Killing it. And then this over here is like an 100 micrometer size. Now, the reason I'm writing out micrometer all the time is because I don't want to get too deeply into that, which is the actual, so if nm is nanometer, then mu m is micrometer. You can just think of it for micrometer for now. If that makes sense to you, then take it and use it. All right. So that's our basic ruler as we start to dig into these different ideas and images of cells. So let's take a closer look at some cell structures, and we'll keep on playing. Recall the cell membrane. Oh yeah. Just to move my clipboard around. I get excited. The cell membrane. Oh my gosh. We're going to keep talking about the cell membrane, and it's going to keep coming back. We're going to get a better and better idea of it. For now, we're actually not going to get too deep into it. All I'm going to do is draw a big chunky cell that looks like this. And I'm going to make a couple decent vague impressions inside of it. So there's a lot of things that we can tell just from an image of a cell. And one of the things that you're going to have to start practicing doing is being able to figure out exactly what the sort of cell is just by the image that you're presented of it. This cell has a number of characteristics that make it immediately apparent. And if you're watching at home, you're probably already aware, if you've done some of this stuff before, what this cell is. If you're not, how could it be that somebody is looking at this and they already kind of know what's going on? And the answer is this guy over here. That guy is a cell wall. Cell walls are present on plant cells. And that's it. We're dealing with a plant cell, right? It's about, what is it? It's about 10 micrometer by about 30 micrometer from what I can tell. So a nice chunky size. There's a lot of really important structures going on inside of this cell, and one of our hopes is that we're going to get closer to it. This rough structure that we can see inside is actually the nucleoid, which is, a part of the nucleus, essentially. Anything else important here? They've also my diagram suggesting to me I point out the cytoplasm, which we talk about all the time. That's the generic name for the internal sort of fluid of the cell. Anything last important, of course. And we've got to talk about the cell membrane itself. So the cell wall and the cell membrane have a very interesting relationship. The cell wall exists only on plant cells, and it's larger and it's chunkier, and it provides a much greater degrees of defense, essentially. And the reason for that is, if you think about it, plants can't escape. Actually being able to move out of a location is a really good defense against bacteria and viruses and a whole bunch of other things. You know, find a new water source or whatever it is. Plants don't have that, so they need some other defense, and this is what they've got. So the cell wall itself then has underneath it the regular old cell membrane. So we have this actual cell wall section, of course, which is this actual thick bit, and then underneath it we have the cell membrane. The cell membrane is our same favorite, permeable guy that we are all familiar with, and we're going to start talking much more specifically very soon in this section of the course about exactly how it functions and what chemicals make up it. Right, well, that's sort of where we're at. So this is a really obvious cell. It's a really obvious plant cell. Let me draw another really obvious cell that is one of the classic ones that everybody's introduced to super early on. I'm going to go for a more sort of ovaloid structure, and then off the tail we're going to get these guys, and then off the side we're going to get these guys. So this is a sort of default drawing of a totally different type of cell. And you can think of this in some ways as a bacteria, right? But bacteria is a huge class of a whole bunch of different crazy things that look in all sorts of different ways. But what we have going on here is something that's appears to be free floating, that lives in water. It's got flagellum coming out of its back so it can swim. It's got some sort of like sensory pili, which is real. These are just like very generic names. The flagellum, propellet, the pili are like sensory hairs, essentially. Pili show up as a generic name for hair-like biological structures all over the place. You'll encounter it many, many times. It's worth knowing as that in a general context, as opposed to just these particular hairs. Bacteria actually belong, they're called prokaryotes. Are prokaryotes. And this is a very specific piece of biological terminology because it refers to the existence of DNA in or outside of a nucleus. So this is DNA without a nucleus, which means that it occurred before the invention of the nucleus, which means it's before the nucleus. So the pro bit means before. A karyotes, it's like true beings or something. I'm sorry, my Greek is failing me. I'm sorry, Mom. Prokaryotes, there we are. But it's definitely before the nucleus. Prokaryotes are, of course, one side. If prokaryotes are one side, then eukaryotes, true beings, I think, something like that, are what we are. And eukaryotes are defined entirely by the fact that they have a nucleus. The nucleus allows for a bunch of really interesting things to happen, but as a general rule, it allows for a level of sort of physical complexity in animals for reasons we don't fully understand. We know that this has to be the case because when we encounter all sorts of crazy multicellular animals, we're dealing with eukaryotes, but we don't understand the exact mechanisms that are important for this to take place. And in fact, one of the things that people suggest as being much more important than just the existence of a nucleus is the role of mitochondria. Mitochondria are really cool. And mitochondria are also a special eukaryotic feature. So, that takes us to this stage. How much time we got left? I got tons of notes to go. We talked about a bunch of really interesting stuff. I want to say probably at least one last interesting thing. Yes. So, One of the things that both of these guys have, plant cells, in fact, all the sorts of cells that we have, is somewhere inside them, they have these little guys called ribosomes. And ribosomes are really important because if we think of these incredibly important structures that we've talked about, these enzymes are basically, they're magical. They have the magical ability to catalyze reactions, which means they can make chemicals. Just at will, right? As long as the body has produced the enzyme that can do it, then the body can make that chemical, which is kind of mind-blowing. Because these enzymes have the ability to make chemicals, the chemicals that they make, that it's most important that they're able to make and that they can make in the body, are more enzymes. But this requires a very particular process, which is called the transcription and translation of DNA. And I know this is all very small right now, but this is super important. We're going to come back to it and do it all in detail, right? So the transcription and translation of DNA allows more enzymes to be created. And the ribosomes actually turn out to themselves be enzymes that are specialized in this process of transcription. Well, in this process of translation. And translation is the turning of DNA information into actual proteins, right? So that hints at a mechanism by which the entire cell can be created out of chemical components. That's where we're going to stop for now. There's going to be a second part to this. Stick around. Have a great day. You're the best. I hope you're enjoying this. Yeah. Have a good one. So we got a picture here, man. We got a picture. Ooh. And this is supposed to be a picture of what comes out of an electron microscope. It's a little hard to tell what's going on right now, so we're going to toss in some labels and hopefully we'll start to figure things out. So one of the basic things that you should probably know is that this right over here is the nucleus. And so we're inside of the nucleus over here, which means this has to be some sort of nuclear membrane. Straightforward stuff. And actually, we have a few holes inside here too. One here and one here. And these are nuclear pores. There have to be nuclear pores in the membrane in order to allow things to flow in and out. Now, we're nice and close zoomed up inside of a cell now, which is kind of fun. We've got a bunch of interesting sort of wavy shapes around here, but first I'm going to focus you on the fact that a bunch of these wavy shapes have interesting little dots on them. These little dots, you can see over here, are actually ribosomes. They're ribosomes. And you remember that ribosomes from the last class play this really important role in creating you. They play this really important role in producing proteins. They're the sort of like cheaty enzymes that our body uses to create all sorts of other crazy enzymes and crazy structures that our body can then do all sorts of other crazy things with, such as breathe or teach cell structure things. Yeah, it's cool. So these guys over here, these interesting little circle structures, are probably mitochondria, but we don't want to focus too much on those right now. The ribosomes are located, they can free float as well, but they're also, they're often located on what we call over here, the endoplasmic reticulum, right? Specifically, if it has ribosomes on it, it's called the RER, the rough endoplasmic reticulum. And the RER is therefore really obvious in terms of this section over here. Got lots of RER over here. If we've got rough endoplasmic reticulum, and it's rough because it has ribosomes on it, R for rough, R for ribosome, then smooth endoplasmic reticulum is the same stuff, roughly speaking, but without the ribosomes. Why would there be both rough and smooth endoplasmic reticulum? So, as a general rule, rough endoplasmic reticulum is adjacent to the nucleus and smooth is sort of further away, which gives you an immediate sort of impression of how because the ribosomes are so important and the DNA itself is so important and the RNA, which takes information from the DNA to the ribosomes, is so important, you want that all protected by additional layers of stuff. So maybe that's what the smooth endoplasmic reticulum does. And that is definitely part of its function here. It's represented on this image SER as so, like that. Now, different molecules, different cells have different proportions of smooth endoplasmic reticulum and rough endoplasmic reticulum, and they also have totally different internal structures. There's tons of glorious variety in the cell world, just as there's tons of glorious variety in the actual macro world that you can see around you with your eyeballs. So, anything left really important I want to talk about here? I think the most important thing we got to start emphasizing, our takeaway, is that the ribosomes receive, and give you another letter, mRNA instructions from the DNA in the nucleus, travels out through, of course, the nuclear pores. And what does those instructions do? They tell you what protein to create. That's, if you could read them, that's what you'd do as well. Ribosomes receive mRNA instructions from the DNA in the nucleus on what protein to create. This is our basic sort of overall takeaway, and this is our image for sort of where this stuff might actually start happening. This is inside all eukaryotes. So in all of your cells, this sort of exciting action is taking place right now, keeping you alive, keeping you happy. These guys we're now going to start talking a little more closely about. This guy 1 and 2 over here, we think these are probably mitochondria. Now, let's take a closer look. If they are mitochondria, let's take a closer look at what they would look like. I'm going to draw a mitochondria here. Now, I usually simplify my markers are getting sad here, guys. I usually simplify my mitochondria to just like this simple, or when I'm doing smaller coffee, I do like a simple little coffee bean sort of shape there, which is both funny and surprisingly accurate. But here we're going to look at, say, this mitochondrion right here. It's located beside a whole bunch of rough endoplasmic reticulum, which I'm going to draw in now in the same sort of style. Make sure we've got our roughness on here so we know that it's rough. The reason in this case that we want it by our rough endoplasmic reticulum is very simple. It's fueling the protein synthesis that's occurring at the ribosomes, which I think is really nifty. So internally, the structure is famous. It's basically this sort of plated structure. It has this internal, very distinctive sort of set of plates. These actually are functional, and they play a role in how the mitochondria generate energy, but I don't want to talk about that right now. That's complicated. All you really need to know as a general fact is that there's a whole bunch of special, it has its own special type of DNA, essentially, and it also it was once its own like separate organism that was sucked inside of us, and as part of that, like several other organelles, actually, it has its own cell membrane as well. Where's my black pen? There she is. There's not the one I want. All right, so it's got its own cell membrane here as well, which is really cool. I'll label my ribosomes again. So the energy, and the energy generated by the mitochondria is actually, roughly speaking, in terms of ATP. The energy that's generated in terms of ATP is then sent to the ribosomes to fuel protein construction. And this is a really good way to start thinking about the location of mitochondria. Mitochondria are typically going to be compared directly, or going to be related directly to the actual thing that they're trying to fuel. Our scales are a little hard to figure out here, so let me try and redraw this a tiny bit. If my nucleus is like this big, then my mitochondria probably about like this big, right? And mitochondria have some variability in size and scale as well. So the nucleus is a much more big, much more complicated structure. And keep in mind, in this particular case, all of this is undoubtedly happening within the great excitement of the endoplasmic reticulum. Endoplasmic reticulum, which again is this sort of housing and protective structure and a transformative information structure that exists right around the nucleus. It's really cool, does a ton of really important things, and to my knowledge is not understood quite as well as some of this other really cool stuff. So this is hopefully starting to get your brain gear in terms of the different basic relationships between all of these cool pieces, between these chemical pieces and these biological pieces in particular. We're going to continue diving deeper and deeper into this and explore more and more stuff, and hopefully it just gets more and more interesting and you guys enjoy it. Thank you very much for watching. Have a great day. Tell everybody in your life that they're awesome, and believe in yourself and all that good jazz. Bye for now.